Absorption by roots. This chapter carries immense weight in your ICSE board exams. You're going to see it frequently in Section A for precise definitions and MCQs, and it routinely anchors Section B, where diagram-based structured questions will strictly test you on root hair adaptations and cellular mechanics like plasmolysis. Listen, if you're aiming for that perfect 80 on 80, mastering the absolute technical precision of this chapter is completely non-negotiable. Here is our roadmap for this explainer. First, root hair structural adaptations. Then, imbibition, diffusion, and osmosis. Third, tonicity and cell mechanics. Fourth, turgidity, plasmolysis, and flaccidity. After that, we'll hit active versus passive transport. And finally, the forces driving sap ascent. Section 1. Root hair structural adaptations. Take a close look at this microanatomy. You'll see these cross sections all the time in Section B. Now, notice how the root hair isn't actually a separate cell. It's just a direct, outward extension of a single epidermal cell. And crucially, it houses this massive central vacuole filled with cell sap. This specific anatomical setup right here is the literal mechanical foundation for all water absorption in the plant. You absolutely must memorize these 3 syllabus-defined structural adaptations. First up, an enormous surface area that maximizes contact with soil water. Second, the cell sap concentration inside that vacuole is consistently higher than the surrounding soil water, which is what establishes the gradient for endosmosis. And third, it has a freely permeable, thin cell wall paired strictly with a semi-permeable cell membrane. Don't mix those up. Section 2. Imbibition, diffusion, and osmosis. We need to draw some really sharp distinctions here. Think of imbibition as the passive absorption of water by hydrophilic or water-loving colloidal substances of the cell wall via surface attraction. It's kind of like how dry seeds swell up when you soak them. Diffusion, on the other hand, is the free movement of molecules from a region of higher concentration to lower concentration, strictly without requiring a membrane. Now, osmosis introduces a critical barrier. It's the movement of water molecules from their region of higher concentration to their region of lower concentration, specifically through a semi-permeable membrane. The presence of that membrane, like the plasma membrane of the root hair we just talked about, is the absolute defining differentiator from simple diffusion. Depending on that concentration gradient, osmosis takes 2 forms. Endoosmosis is inward diffusion through a semi-permeable membrane when the surrounding solution is less concentrated. That causes the cell to swell up. Exoosmosis is outward diffusion when the surrounding solution is more concentrated, and that directly causes cellular shrinkage. Notice the mechanics at play in these diagrams. With endosmosis, water is forced into the central vacuole, swelling the cell until it's fully distended. But with exosmosis, water is forced out of the cell sap, physically shrinking the protoplasm away from the cell wall. You need to know these exact visual mechanics for the exam because they are highly testable. Section 3. Tonicity and cell mechanics. Tonicity essentially describes the surrounding medium relative to the cell sap. So, a hypotonic solution has a lower solute concentration outside, which drives endoosmosis and cell swelling. A hypertonic solution has a higher solute concentration outside, triggering exosmosis and cell shrinkage. And an isotonic solution, that has an equal concentration, yielding a dynamic equilibrium where there's zero net movement of water. Section 4. Turgidity, plasmolysis, and flaccidity. When endosmosis pushes a cell to its absolute physical limit, it becomes fully distended, or turgid. Turgidity happens when hydrostatic turgor pressure, which is exerted by the swelling vacuolar cell sap, pushes outward, but it doesn't burst. This outward push is perfectly and rigidly countered by an inward wall pressure from the cell wall itself, keeping the plant structurally intact. Conversely, if you drop that cell into a strong hypertonic solution, it rapidly loses water. Look at the extreme state in the final diagram here. That's plasmolysis. This is the explicit physical contraction of the cytoplasm and the tonoplast away from the cell wall. A completely plasmolized cell is said to be limp or flaccid, which is the exact mechanical opposite of turgidity. Section 5. Active versus passive transport. So passive transport is the free movement of molecules along a concentration gradient, meaning from high to low, without spending any energy. Active transport, however, is a whole different ballgame. It's the passage of a substance from its lower concentration to a higher concentration through a living cell membrane. Doing this strictly requires metabolic energy in the form of ATP. Check out the carrier proteins embedded right there within the living cell membrane. Here's the thing. Vital nutrients like nitrates, sulfates, and potassium are often in lower concentration in the soil than they are inside the root. So the cell simply can't rely on diffusion. It has to expend ATP energy to forcefully pump these critical ions against the concentration gradient and into the cell sap. Section 6. Forces driving sap ascent. This massive upward journey begins at the root hair, which absorbs water via endosmosis. That triggers a chain reaction of sequential cell-to-cell osmosis right through the cortex tissues. As one turgid cell presses against the next, a strong inward hydrostatic pressure builds up. Ultimately, this pressure literally forces water into the thick-walled xylem vessels, generating a foundational upward push known as root pressure. But root pressure alone isn't going to push water all the way up a massive tree, right? The complete ascent of sap actually relies on 4 synergistic forces working together. First, you have root pressure, providing the initial inward push. Second is capillarity, which is the physical rise of water within extremely narrow xylem vessels. Third is transpiration pull, a massive suction force generated by aerial water loss. And finally, cohesion and adhesion forces, which work together to maintain a continuous, unbroken water column all the way up. Gain a definitive edge in your ICSE Class 10 board preparation. Subscribe to our YouTube channel at ICSE Masterclass 10 for complete, zero-fluff conceptual clarity and step-by-step diagram breakdowns. To download your copy of the premium handwritten biology notes, worksheets, and past year's question analysis used in this session, immediately join our official Telegram community at https.telegram.me/ICSE Class 10 WPIV. Before you go, consider this. If transpiration pull is the absolute strongest force lifting water, what exactly happens to a towering tree on a completely humid, windless day when transpiration just stops? Think about that for a minute, and we'll see you in the next explainer.